Okay, so in this video, I'm going to go over chapter 15, which goes over biosynthesis. So now that we've talked about how bacteria kind of produce energy, now we're going to talk about how some bacteria produce molecules or kind of put molecules together. So we were previously talking about how we get glucose and break it down. Now we're kind of talking about the other way, uh, making things. Okay. So we're gonna do a quick overview of biosynthesis, and then of course we're gonna talk about the Calvin cycle, which is the main way that bacteria make things. Um, and yeah, and of course you probably learned about the Calvin cycle when you learned about plants and general bio, because that's what plants do after photosynthesis. Is they use the energy that they make in photosynthesis to then put carbons together to make glucose. Okay, and then in 15.3, we're going to go over how bacteria produce fatty acids. We'll also talk about how they produce or fix nitrogen and make compounds that are made up of nitrogen. So those are things like amino acids and nitrogenous bases, which are your DNA and RNA molecules. So, okay. So this is kind of an overview of what we're going to be talking about. And as you can see, a lot of what we're going to be talking about is how bacteria produce glucose, but we'll also be talking about how they produce things like lipids, fatty acids, amino acids, things like that, peptides as well. So this is all kind of one giant overview. One of the main processes that bacteria can do is this process called gluconeogenesis, which is basically them producing glucose. And usually this is done by doing reverse glycolysis. So if you remember, glycolysis is when you get glucose and you break it down to make pyruvate. And so bacteria are able to go backwards, get pyruvate, put it together, get two pyruvates, put it together to make glucose. So we'll talk about that also today. Okay, so obviously this is like a really huge process, and I'm not going to make you memorize all of this. But I just kind of wanted to show you how the TCA cycle is kind of central in a lot of biosynthesis in bacteria. So the TCA cycle is not just to get glucose and break it down, and then that glucose produces energy. So the TCA cycle has a lot of other things. So here again is the TCA cycle. Remember that we get citrate. As a citrate, there's two decarboxylation steps where there's CO2 being released, and then it goes through the cycle. Now, a lot of the compounds that are part of the TCA cycle are used to make a whole bunch of other stuff. So for example, oxaloacetate, which is what combines with acetyl-CoA. Oxaloacetate by itself can actually be converted into aspartate, asparagine, and a bunch of other things, and a bunch of other amino acids essentially, and also nucleic acids, the pyrimidines. So that's one really important one. Succinyl-CoA is also used to make a whole bunch of stuff like vitamin B12, and again a bunch of amino acids. Down here, alpha ketoglutarate or 2-oxoglutarate. This is used to make, again, a bunch of amino acids, right? Glutamate, glutamine, blah, blah, blah. So we'll talk about that. And then acetyl-CoA. So if the acetyl-CoA doesn't go into the TCA cycle, it can actually be used to make a whole bunch of other stuff. And so as you can see here, the acetyl-CoA can be used to make lipids and leucine, pyruvate. So if we went backwards one more, pyruvate makes a bunch of other stuff. And then all of the little steps, so this is glycolysis here, all of the steps of glycolysis and also the pentose pathway, remember that these are also used to make a whole bunch of other random stuff as well. So these processes that we learned in the last couple chapters are not just for energy production, they are also for making other compounds. So bacteria have a lot of different ways to do things. Okay, and so before we go into all of the crazy unique things that bacteria can do, let's go over the Calvin cycle, because the Calvin cycle is the main cycle that makes glucose, and even plants do this. So this is kind of a universal way of making glucose that both bacteria and plants share. So hopefully you sort of remember how this process works. We're basically taking in carbon dioxide and fixing it. That's why it's called carbon fixation. And we're going to make a 3-carbon molecule called G3P, or glyceraldehyde 3-phosphate. So this is the main compound that we're producing at the end. So how does it all happen? The first molecule that's being used is ribulose 1,5-bisphosphate. So maybe you've recognized this, but this is the first compound that combines with CO2, and then it makes PGA. So it makes a 3-carbon molecule, but 2 of them. So basically this is 1, 2, 3, 4, 5 carbons, and we add another carbon from CO2, and then it splits in half. So now we have 2 molecules that have 3 carbons each. Notice, by the way, that they're saying 3 and 6. The reason why is because when you complete the Calvin cycle, you actually go 3 times around. But I just want to keep it simple. 1 of these combines with 1 CO2 to make 2 of these PGAs. And then eventually, at one point, we make 2 glyceraldehyde 3-phosphates. So that's really the main thing that we're going to produce, G3P, and G3P will leave the cycle and will eventually be glucose, will be converted into glucose. So 2 of these G3Ps is used to make glucose. Remember that glucose has 6 carbons, so you need 2 of these guys to make the 6-carbon molecule. Okay, and again, I don't want to go into too much detail about how everything happens. It's just you need to know ribulose 1,5-bisphosphate, and G3P is the end product that we really care about. Now, the enzyme that does this part combines the ribulose 1,5-bisphosphate with the CO2 is one of the most important enzymes on Earth. It's called Rubisco. And yeah, this is how it does it. You don't really need to know all of this, but it's the enzyme that combines the 1,5-bisphosphate with the CO2. And so it's showing how it does it. So it sticks it onto here, but then the CO2 is unstable. So then we hydrolyze it and it splits it into the PGA, yeah. So that's what that process here, from here to here, this is like a zoom in of what happens. So you have a 5-carbon molecule with another CO2 being added. So now we have a 6-carbon molecule, but it's super unstable. So it splits into PGA. Yep, that's pretty much that. And Rubisco, again, is super important. It's one of the most abundant enzymes on Earth because we find it in all plants, but then also in bacteria. Okay, so this is the classic, method of producing molecules. But bacteria also have a bunch of other special processes that they can do. And so one of them is the reverse TCA cycle. So remember, we just talked about the TCA cycle. Some bacteria can go in reverse. And the reason why is because all of the arrows in the TCA cycle are actually reversible. So they can go in one direction or the other. And so the reverse TCA cycle basically takes in CO2 instead of releasing it. So instead of getting citrate and breaking it down and then getting a CO2 out and then taking another CO2 out, we're going backwards. So we're starting with a 4-carbon molecule and then we're adding a CO2 and then we're going to add another CO2 and then eventually we'll get to glucose. So yeah. So how does this work? Where should we start on this picture? I'm just going to start with oxaloacetate. It's just easier. So we're here with oxaloacetate. It gets converted back to malate, fumarate, succinate. So everything is going backwards. And literally everything is being used up. So now we're using ATP. So remember, if we were going from here to here, remember it produces ATP. So now we're going backwards and we're having to use ATP to do this. So now we're in succinyl-CoA, and then we're going backwards again. We're going to make alpha-ketoglutarate. And in this point, we're taking in a CO2 molecule. So we went from 4 carbons to 5, and then we're going to go one more. We're going to go from alpha-ketoglutarate and produce isocitrate. And again, we're going to take in a CO2. So now we should have 1, 2, 3, 4, 5, 6 carbons. So now we're at the 6-carbon molecule, which is good. That gets converted into citrate, which is, remember, why it's called the TCA cycle. The citrate will get converted back into acetyl-CoA, which, guess what, gets converted back into pyruvate. And then from pyruvate, it goes into the gluconeogenesis, which is basically reverse glycolysis. So all of this acetyl-CoA to pyruvate to glucose is the gluconeogenesis part. So yeah, pretty interesting. And then, of course, the pyruvate can be converted into phosphoenol pyruvate, which, guess what, can be made into oxaloacetate. And so we have like a little bit of a bypass or like a figure 8 in this process. So it's pretty crazy. Anyway, so bacteria can do that. So it's literally the reverse of the TCA cycle. There's nothing else to say about that. So all right. Another pathway that bacteria can do is called the reductive acetyl-CoA pathway. So they're getting they're producing acetyl-CoA by reducing. So we get CO2. Okay, so it's getting CO2 from the air, the bacteria, and they're going to reduce it to this compound called formic acid. So basically they get CO2 and they combine it with some hydrogen and they form formic acid. Okay, that's weird already. And then that formic acid gets converted into this eventually it will be converted into this TbCH3. So you don't really need to know all of these random steps, but basically the formic acid will eventually be converted into this molecule here, TbCH3. Okay, weird. And then in another part of the process, we get a CO2, another CO2, and we are going to convert it into CO. As you can see here, CO2 is converted into CO. All right, weird. And then together, this CO is combined with the TbCH3 from earlier, and together they're going to make acetyl-CoA. Of course, we have to add the CoA there, but yeah, so all of this and this combine to make acetyl-CoA, which is super weird. And then this acetyl-CoA can go into gluconeogenesis, which produces glucose, or the acetyl-CoA can be used in the TCA cycle, either way. But yeah, kind of a weird way to make acetyl-CoA. A lot of people remember or think that the only way to make acetyl-CoA is from pyruvate, but actually you can make acetyl-CoA, well, bacteria can. They can make it from CO2. So they just get a CO2, another CO2, and they do both of these pathways to make acetyl-CoA. So super weird. Okay. Awesome. So another kind of pathway that they have, I'm putting a smiley face here because really it's like a lot. I'm not going to make you memorize all of this, but this is just one way that they can make fatty acids. So the main thing that you really need to know is that the beginning of fatty acid biosynthesis starts with acetyl-CoA. Okay, acetyl S-CoA, by the way, but whatever. So acetyl-CoA is combined with CO2 to make this weird molecule, manolyl-CoA. So it's a 3-carbon molecule. And then it gets converted into manolyl ACP, which is also very weird, but blah, 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 a bunch of random stuff happens. And then as you can see, we're just starting to create this chain of carbons. So remember that a fatty acid is really just a hydrocarbon, right? Carbons and hydrogens in chains. And so as you can see here, once we start producing this, we just keep adding more and more carbons, and then it becomes your fatty acid chain. And you can have multiple, different sizes. So, but really the thing that you need to know is that the beginning of it is acetyl-CoA. Okay. Bacteria obviously can do a lot of stuff with nitrogen as well. So they can fix nitrogen, they can convert it into pretty much any form. And so we're really going to be focusing on this part of the diagram because this part of the diagram we've already kind of talked about in the class. So I'm really just focusing more on this. And the two processes. So ammonification basically is how bacteria can get proteins and nucleic acids and break it down into ammonia or ammonium. Whereas the assimilation process is the opposite. They will get ammonium and they can convert that ammonium into proteins, nucleic acids, and other things. And so bacteria can go back and forth. But this is a pretty cool process. They can basically get this ammonium, which, by the way, they can just get it from, if you're a soil bacteria, there's a bunch of ammonium in the soil. Or if you are a nitrogen fixing bacteria, you can get N2 gas, convert that N2 gas into ammonium, and then that ammonium is used to make your proteins and nucleic acids. So they have a lot of ways to kind of survive with very little resources if they can just take the nitrogen straight from the air. So that's pretty cool. Okay, the protein that does this process, by the way, going from N2 to ammonium, so the nitrogen fixation process, the enzyme that does this is called nitrogenase. So the nitrogenase converts N2 to NH4. And the enzyme is a pretty weird looking guy. It's got like an Fe unit and an FeMO unit. It's got two weird units. Again, you don't really need to know this entire process, but I'm just kind of going to follow through with this diagram real quick. So basically, this is the complex or protein, and it's going to take in an N2. Remember that N2 is N triple bonded to another N. That's the gas, right? And then it starts, of course, this whole process uses a bunch of ATP. Notice that it's like 4 ATPs just for all of this. But basically what it's going to do is start reducing the nitrogen and adding electrons to it. And when it adds electrons, it also adds hydrogen so that it stays neutral, right? Electrons are negatively charged, so that has to add protons to make it positive or neutralize it. But anyway, as you can see here, electrons are being added. So now it becomes an N double bonded to an N with two H's on each side. Cool. And then it does that again. So now we have an N single bonded to an N with an H2 and an H2. Okay, and then down here, eventually we get, I really don't, oh, I think this is like, okay, yeah, I'm skipping some stuff. So it's like, this diagram is like going in a zigzag, but we don't really need to know all the steps. Just know that we keep adding hydrogens. And so now we're here, and then eventually we get to NH3. And then guess what? We stick one more hydrogen on there and we get NH4. So it's just progressively adding hydrogens to the nitrogen until it becomes NH4. That's pretty much it. Okay, so that's this enzyme, nitrogenase. Nitrogenase is this enzyme. Now, bacteria that can do nitrogen fixation, so they have this enzyme, they regulate this process very tightly. And the reason why is because the nitrogenase, the enzyme, is very sensitive to oxygen. If oxygen is present, this usually doesn't work. And the reason why is because oxygen is a very strong electron acceptor, right? And notice that we're playing with electrons here. So if oxygen is present, it might just suck up all the electrons. And that's, obviously not good. Right? We're trying to reduce the nitrogens here, but the oxygen will just steal all these electrons. So bacteria, when they're trying to do nitrogen fixation, they'll do it only when oxygen is really low, because if oxygen is super high, this enzyme's not going to work anyway. And then bacteria regulate their nitrogenase depending on if NH4 is present, right? So this whole enzyme's purpose is to produce NH4. Now, if there's already a lot of NH4, there's no point in making this enzyme. Notice how much ATP is being used. It's a very costly enzyme and a very costly process. So if there's already an abundance of NH4, there's no need to be making this enzyme. So we're going to talk about the regulation of this nitrogen fixation. So the enzyme that makes nitrogenase is this whole thing right here, NIF-HDKTY. So NIF-HDKTY is the gene that produces the nitrogenase enzyme that we just saw in the previous diagram. Now, there are a bunch of other proteins, regulators, that work together to regulate whether this gene is on or off. And that all depends on the amount of NH4 that is present and also the amount of O2 that is present. Okay, so for example, if we have a high NH4, then there shouldn't be any nitrogenase being made, right? So we shouldn't be producing this because there's already NH4. And so how does that work? Well, the first step in turning on the nitrogenase gene is to convert this NTRC into NTRCP, meaning that we just added a phosphate group to this protein. So this protein called NTRC needs to be phosphorylated, and that is the first step turning on the nitrogenase gene. Now, this phosphorylation process only happens when there's low NH4+. So if there's high NH4+, then this first step never happens. So this whole process just doesn't even exist when there's high NH4. So the nitrogenase is not made. Makes sense? So if there's low NH4, then the NTRC is phosphorylated. Okay, so we have NTRCP, and NTRCP, the phosphorylated version, goes over here, and it basically turns on these two genes, NIF-L and NIF-A. Okay, and NIF-L and NIF-A are super important because they're going to be controlling the nitrogenous gene. They're going to be turning it on or off. Okay, cool. So that's that part. So low NH4 is what turns on NIF-L and NIF-A, and those two things are going to be important when we need to turn this gene on. Now, remember I told you that we also need to have low oxygen levels, because if there's high oxygen levels, then the nitrogenase enzyme doesn't work anyway, so what's the point? And so that's the whole point of NIF-L and NIF-A. They're able to turn the gene on or off depending on the oxygen levels. So if there's high oxygen, we don't want the nitrogenase from being made, because again, nitrogenase is useless when oxygen is high. And so what happens when there's high oxygen is NIF-L will bind to NIF-A. So it will bind to it, so they will produce this giant complex together. and that will prevent NIF-A from binding here. NIF-A happens to be the activator for the nitrogenase gene. And so if NIF-A is being occupied or being blocked, not really blocked, but NIF-L is basically preventing NIF-A from even binding, then the gene doesn't turn on. Okay, so even if we had high NH4, or sorry, even if we had low NH4, right, meaning that we turned on the NIF-L and NIF-A genes, if there is high oxygen, NIF-A will not be able to turn on the nitrogenase gene. So there's like two levels of regulation because, again, we want to make sure that there's low NH4 and we want to make sure that there's no oxygen or low oxygen. So when there's a low oxygen environment, then NIF-A is not attached to NIF-L, meaning NIF-A is free. And NIF-A that is free is going to bind to the binding site here, and that basically turns on the transcription of the nitrogenase gene. The other player that's involved that I didn't really talk about was sigma 54. Sigma 54 is also involved. It binds to both the NIF-LA operon and also binds to the NIF-HDKTY gene or operon. So it's involved in both here. So you also need sigma 54 to be present for any of these things to happen. But I didn't really want to focus on that. I really wanted to focus more on NTRC and its whole job, and NIF-L and NIF-A. Okay, and by the way, don't worry about NTRB. NTRB, the only function was to phosphorylate this guy, so that's fine. All right, so that is how it's all regulated. And so again, bacteria want to only turn on the nitrogenous gene if there is low NH4 and also low O2, right? If there was high O2, then it still doesn't turn it on. And if there was high NH4, then obviously it doesn't turn it on either. Okay. Moving on to amino acid biosynthesis. So again, this is like a lot, so I don't really want to go into too much detail, but as you can see, the central pathway that is required for most amino acid biosynthesis is the glycolysis pathway and then the TCA cycle. So you should recognize that this is glycolysis because we're going from glucose to G3P and then eventually pyruvate, right? So you should recognize all of these pathways. And as you can see, a lot of the molecules that are made during glycolysis are used to make amino acids. So the 3-phosphoglycerate is used to make all of these guys. Phosphoenolpyruvate is used to make a bunch of these. If we went into the pentose phosphate pathway, we would make a bunch of these guys, right? So there's a lot. Pyruvate makes a lot. Valine, leucine, alanine, lysine. And remember, I showed you that oxaloacetate is responsible for making a bunch of amino acids, and alpha-ketoglutarate, or also known as 2-oxoglutarate, this makes a bunch of amino acids also. So a bunch of things that make a bunch of other things. So the main ones that you really should know are oxaloacetate making these amino acids. You don't need to know that it makes these specifically, but you just need to know that oxaloacetate and alpha-ketoglutarate are two of the main ones in the TCA cycle that make amino acids. Pyruvate is also another big one. And phosphoenolpyruvate. Yeah. Those are like the four main ones. This one, and that one. Okay. And as you can see, making amino acids is really rough. And for bacteria, they have to prioritize which amino acids they're going to use. And so, for example, if you make a protein and that protein remains inside of the cell, when that protein gets degraded, all of those amino acids get recycled. That's how the bacteria work, is when they use their proteases to degrade proteins, like at the end of the protein's life, those proteins, the amino acids get recycled. Now, if you are making an external protein, meaning a protein that is outside of the cell, you basically will not be able to recycle those proteins because now they're outside, right? It's not like you're going to take them back inside to recycle them. And so you'll notice, especially for the flagella, that all of the proteins that are involved with the external part of the flagella are less expensive than the parts that are internal, because it wants to, again, conserve the expensive ones. So Here, all of the proteins that are used to make the external part of the flagella, as you can see, are in the orange or yellow, which are extremely expensive amino acids. But you'll notice that all of the proteins that are inside, or proteins that are kind of in the cytoplasm hanging out, those are going to be less expensive. And the expense has to do with how much energy it took to make those amino acids. So as you saw here, there's a bunch of different ways to make amino acids, and some of these are more expensive, quote unquote, than others, meaning it takes more ATP, it takes more ATP or resources to do it. So yeah. Okay, one amino acid that we're going to focus on is glutamine, how glutamine is produced, and also glutamate, so glutamate and glutamine. So there's actually three ways, or three different pathways, I guess, that we can produce these two molecules. So alpha-ketoglutarate is really the central part. And actually, sorry, I keep going back and forth. You'll see here that alpha-ketoglutarate, which is also known as oxoglutarate, is used to make the glutamate and the glutamine, right? So you can see that here. So that's what we're focusing on. So AKG, which is alpha-ketoglutarate, okay, can be converted into L-glutamate using this enzyme called glutamate dehydrogenase, or GDH. So that requires energy. Notice that it takes up an NADPH when it does it. And then, yeah, we make glutamate. Cool. So that's that. Or another thing that can happen is that glutamate can be converted into glutamine using glutamine synthetase. Okay. Cool. Or let's say that we really want to make more glutamate for some reason. There's another pathway. You can get alpha-glutarate and glutamine, so both of these molecules at the edge, and you can combine both of them to make two molecules of glutamate. That's why it has a two molecules here, because this plus this can also make more glutamate, which is super weird. And that is through this glutamate synthase, or GOGAT protein or enzyme. So yeah, different ways to make glutamate or glutamine. There's like different pathways for all of them. And if you had glutamine, but you needed more glutamate, you can just combine it with more alpha-ketoglutarate to make more glutamate. So yeah, kind of weird process. And one thing that, as you probably have guessed, is that bacteria regulate this process super specifically as well. And so in the next slide, we're going to specifically talk about GLNA, which is the gene that produces glutamine synthetase. Okay, so glutamine synthetase is the enzyme that converts glutamate into glutamine, and it uses ATP to do this. And we're going to talk about the regulation of this gene specifically. Okay, so here it is, the regulation pathway. So I know it looks like a lot, but we're gonna just focus on a couple things. And notice that the diagram is split into low glutamine and high glutamine, because again, this gene, GLNA, is what makes the GLNA protein, and this protein converts glutamate into glutamine. Now, if we had low glutamine, we want this to be active. But if we had high glutamine, we obviously want to turn this off because we don't need any more glutamine. So let's talk about what happens during low glutamine. So during low glutamine, what's going to happen is NTRB, NTR, oops, I did a typo because I put NRT, sorry. Let me fix that. That should say NTRBP. Okay, and this should also be NTR, sorry. NTRC, okay. Anyway, so NTRB is going to phosphorylate, get phosphorylated, and then that also phosphorylates NTRC. Okay, so all you really need to know is that both NTRP and NTRC get phosphorylated. So there we have a molecule that is phosphorylated, this NTRC. Okay. Now NTRC phosphorylated, the version that is phosphorylated is going to go bind here, and it's going to turn on the GLNA gene. It's basically the activator for this gene. Okay, simple enough. Now that is just turning on the gene, but we also need to maintain the activity of the gene itself. Remember that this gene, or this protein, uses ATP to produce glutamine. Now, when it produces ATP, the ATP gets converted into AMP. And if that AMP is stuck on the GLNA the whole time, it's inactive. So we have to remove the AMP, essentially, is what we have to do. Anyway, so to keep the protein itself active, we have another set of proteins that are involved, which are down here, GLNB. And GLNB basically is inactive unless it has a UMP stuck to it. So UMP is uracil monophosphate. So when GLNB is added to UMP, then it can go to the GLNA and remove that AMP. Remember that AMP, I'm just saying a bunch of letters. You're like, what? So remember that when the GLNA protein is attached to AMP, it's like basically turned off. So here's a picture of that. GLNA with AMP is the inactive form. Now, when the GLNB is attached to UMP, it is capable of removing this AMP to make the GLNA the active form again. Okay, so it basically maintains the activity of GLNA and makes sure that there's no AMP attached to it. Okay, so there is this beginning part is the regulation of the gene, and then this end part here is the regulation of the protein activity. Okay, so gene regulation and protein activity regulation. Okay, and so that is what's happening when there's low glutamine. Now, if there's high glutamine, what happens is NTRB and NTRC are not phosphorylated. It just doesn't get phosphorylated. And so if it doesn't get phosphorylated, we never make GLNA, right? That's the whole point of that. But on the protein regulation side, the GLNB is, it doesn't have the UMP. And so if GLNB does not have UMP, it cannot convert the inactive form of GLNA into the active form. So that's the protein side of the story. So remember that for this regulation, there's gene regulation, and then there's protein activity regulation. So two kind of sides of the story. So the NTRC protein is what regulates the gene side, whereas GLNB is really what regulates the protein side. NTRC can only turn on GLNA, the gene, if it's phosphorylated, whereas GLNB can only activate the protein side of things if it has this UMP. So basically, these proteins by themselves are inactive unless they have their phosphate or UMP, respectively. Okay, kind of weird. All right, I know there's like a bunch of other biosynthesis pathways. I'm not really going to ask you about this, but this is how arginine is biosynthesized. Arginine is another amino acid. As you can see, it's like really crazy, but it starts out with glutamate. Glutamate gets converted into a whole bunch of random stuff. Ornithine, and then blah, blah, blah. Oh my gosh, glutamic acid, citrulline, arginosuccinate, and then finally it becomes arginine. Okay, I really won't ever ask that because this is like really crazy. Okay, and then for the aromatic acids, there's, again, a really crazy biosynthesis pathway. So really, I guess for the ones where I have a little smiley face, how many are there that I have a smiley face? Because I know it's like impossible to know all of them. Yeah, for all the ones with the smiley face, you really just need to know the beginning. Okay, what's the starting compound that will eventually make that? So here in fatty acid biosynthesis, it's acetyl-S-CoA. For amino acid biosynthesis, like I said, you should just know the main guys: oxaloacetate, alpha-ketoglutarate, pyruvate, right? So, and then for arginine biosynthesis, you really just need to know that it starts with glutamate and eventually gets to arginine. And then for the aromatic amino acids, so those are going to be your phenylalanine, tyrosine, and tryptophan, then it starts out with essentially phosphoenol pyruvate and erythros 4-phosphate, which this one specifically is produced by the pentose phosphate pathway. And so that's like the unique thing there. Yeah. It's like a lot. As you can see, there's like a lot of steps that they're skipping to. Okay. Wow. And then the last one, nucleic acid biosynthesis. So again, super like, there's a lot. But I guess you should know that the purines, okay, purines are going to be your A and G nucleotides, right? So adenine and guanine. And so those are going to be starting off from ribose 5 phosphate, okay, which again comes from the pentose phosphate pathway. That's one way that bacteria can acquire this. So it starts out there, and then a whole bunch of random stuff happens, and then you get AMP and GMP. Okay, whereas the pyrimidines, which are your C and T, okay, those are going to be the produced from aspartate is the kind of original molecule that eventually gets converted into the C and T nucleotides. Oh my gosh. Okay, cool. So yeah, that's all you really need to focus on when it comes to the ones where I have like a little smiley face, because it's like, I'm not going to ask you to remember all of those pathways. All right, cool. Thanks for watching, and I'll see you in the next video.